once again, thank you. And now I invite Dr. Bal Krishna Joshi from Nepal to share with us information about agrobiodiversity measurement using R. Thank you. You've already shared your screen to all the participants. Thank you.
Yeah, thank you, Dr. Christina. Yeah, I'm very glad to share some of the use of art to measure the agrobiodiversity. And I'd like to thank Grassroots Institutes. I'll not take too much time. We know R. R is the very giant free software commonly used nowadays by many scientists, researchers. But if you see the literature, it has not been commonly used to measure the agrobiodiversity. Therefore, I'm giving here some flavor of R, how we can use R to measure or to quantify the agrobiodiversity. These are some topics. One interesting I would like to highlight, agrobiodiversity statistics. We generally study the statistics, but there is a very few content of agrobiodiversity. And I'll describe how we can estimate some of the diversity using different packages. It is a very long process, and we generally in Nepal also organize the training on these topics, and it almost took 3 or 5 days. So I'm here briefing within 30 minutes. I have shared some publications, references, and all the audience can have in details. Some of this information is already presented by all your faculty members. Some may be repeated, and I'll try to escape. The total agrobiodiversity, particularly in Nepal, we have divided into 6 components: aquatic, insect, microorganisms, crop, forage, and livestock. And within each component, there are 4 sub-components: cultivated, semi-domesticated, wild relatives, and wild edibles. And different kinds of measures or methods we can use to collect the data. And here are some lists. And this list were based on our experience in Nepal. We organize the diversity blog, diversity fair, transect work, and others. Total, based on different statistical tools, we can group based on the useness or the objectives of our research. And in totality, we can call the agrobiodiversity statistics. That includes agrobiodiversity measurement, characterizations, classifications, evaluations, and relationship, and including genetic parameters. Earlier, we all know the levels of diversity. I have here further divided the total diversity. We can estimate it's 5 levels, different 5 levels. That includes species diversity, varietal diversity, and within varietal, we may have genetic diversity, including allelic diversity. And considering the types, we have 5 types: functional diversity, morphological diversity, molecular diversity, use value and nutritional diversity, and food diversity. All these different types and levels, we can estimate the agrobiodiversity. Agrobiodiversity here, I will talk about the indicators, the total indicators of agrobiodiversity. Indicators may be at household level. If you are gonna estimate the indicators based on household level, there are many options, and we have divided these indicators into score and another one is index. I'll explain the index later. For household diversity score, there may be, for example, number of species, we call speciesness, in each of 6 agrobiodiversity components, and number of land masses, land type, functional diversity. These all are related to the number, the counting number. That's why we call the score. We have total here 12. We may have more. And another indicators is we call household diversity index. And this index can be measured based on 4 different criteria. For example, one based on species within agrobiodiversity group. And the index may be Shannon diversity index, Simpson index, and species richness. Similarly, the index may be based on landness within agrobiodiversity group. For example, Shannon diversity index, Simpsons, and landnesses. And similarly, these 3 index can be estimated based on landnesses within a species. We can use different criteria to estimate the index. And the total, if we sum this index, we say household agrobiodiversity index, which means sum of from 1 to 9. Similarly, the indicators may be at village level or community level or any site level, or even at the district or province, or even in the country level. And these are the same as household index, as we estimated score and index as an indicator at household level. We can estimate both score and index similarly at village level. These are the same, and some may be different because within a village, there may be many houses, and we can estimate the village level information at an average of each household. For example, average agrobiodiversity household score, average social agrobiodiversity household score, like that. Similarly, we can estimate village diversity index based on 3 different criteria. One, for example, based on species within agrobiodiversity group, and the index are same as the household index. That means Shannon diversity index, species index, evenness, and Simpson index. And similarly, these 3 index can be estimated based on landmasses within agrobiodiversity group. Group means there are many groups. I have already explained, and I have listed these groups. There are 21 groups. Some of examples are cereals, pulses, spices, livestock, summer fruit, like that. And similarly, we can estimate these 3 indexes based on landmasses within a species. Within a species, we may have many different landmasses, and this based on this landmass data, we can estimate these landmasses. And similarly, if we sum up this 1 to 9, and we can say village agrobiodiversity indexes. And other indexes we can estimate, for example, agrobiodiversity index of household, community, district, province, ward, using different kinds of criteria, for example, number of species or land masses. And we can also estimate the analog site index. We also organize one international training on developing the climate resilient variety. There we call the climate analog tools. These tools helps to identify the analog site of the reference site. Based on these findings, we can have analog site index for particular species or land masses. And similarly, we have different kinds of drivers, and the drivers may have different impact on losing or increasing the agrobiodiversity. Using these drivers, we can estimate different kinds of driver indexes as well. For estimating this score and indexes, we can collect the data, including both qualitative, quantitative, and qualitative. These are the total data structure that we may have from the agriculture field with regard to agrobiodiversity. And data types may be agromorphological, genotypic, including both qualitative and quantitative. This can be measured either individual or from representative sample or at population level. And this data may be either spatial or temporal. For diversity index, we generally use qualitative data, but if we have a quantitative data, we can convert them into different kinds of group. We can say qualitative group. For example, we may have different kinds of phenotypic classes, and based on the frequency data on the gene banks, we can also estimate different kinds of indexes. We generally generate data either on stations or on farm or lab experiment, and most commonly used tool is survey data because we collect perception data from the long history or long experience of many farmers. And there are many online survey tools that helps to collect data directly from the field in electronic media. Some of are SurveyMonkey, Google Forms, Sogo Survey, Yamwater Portal, Field Lab, Fieldbook, and some of them are free and we can use freely, and it is very helpful. We are using YAM Portal currently and Google Form and SurveyMonkey in Nepal. And for measuring the agrobiodiversity, the measure objects are household. We can compare, we can estimate the different kinds of index among the household and compare among them. And the objects may be village level or community or site or even agroecozones. The objects may be district, ward, province, or country, or maybe agrobiodiversity component that I saw in the first slides, and in groups, different kinds of crop groups, cereals, grain, legumes, livestock, etc., and species or landmasses, or any particular traits or functions. We can treat this trait as object, and that we can estimate the index or indicator for this particular traits. Now we can see different kinds of software in Google, or just you can Google this software. And I have listed here most commonly useful software for agrobiodiversity data analysis. That means this software can be used in agrobiodiversity statistics. My suggestion and request, because we have here many students, we have to write some kind of textbooks for agrobiodiversity statistics, and it is better to start learning and teaching these courses. And these are the software that we can use to estimate different kinds of parameters that we usually consider in agrobiodiversity statistics. Some of them are free, but some are not. Among this software, the R and R Studio is nowadays very popular because it is giant, dynamic, and many disciplinary persons can use this software, and it is also free. And within this R package, R software, there are many packages that we can use for agrobiodiversity statistics. And most common one is Biodiversity R and Vegan, and other are listed here. And if you want to use, you can explore these ones. I'll briefly describe about Biodiversity R and Vegan. I know you are familiar with R and R Studio. R Studio is more friendly to analyze the data and even to estimate different kinds of indexes. And we have a 4 window in the R Studio. This window we call R script editor, that you can write here any commands that you can edit and save and you can use in later. And another window is R console commands. Here you can see the results or the response or interface from the computer. And here you can see another window that helps to import data directly, because we usually prefer to have a menu driven software, and this is on that is based on menu driven. And here is another window that have a files, plots, and package. Any package, if you want to install, you just click this package and you need to write the package name and you can easily install. And for file preparation, this is the most important task. If you import the data file properly, and you will get the result. Otherwise, if data is not properly imported, the results might be the wrong or different ones. So therefore, for the data, for the diversity index and score, we may have a community data matrix or household data matrix. And this data matrix, my suggestions is to prepare in the Excel. Excel is very flexible and easy to handle and to manage the data. And then we can directly import this Excel data to our studio. After preparing this data file, you can import the data file in two different ways. There are many options, but most commonly used option is you can import directly from the menu using R Studio. Sorry. During this, you need to check column and row names. We may have column and row names, and you need to import both names. And the names may not be considered as the data file during the analysis. These are the labels or text. And for this, you can also write the command. These are the command directly in the console, and you can get the data in R Studio. This is the vegan packages, and it is very popular for ecological diversity analysis. And some function of this vegan we can use for estimating different kinds of indices, particularly richness, Shannon index, and Weber index. This is very simple. These are the command. The yes, if you write this after importing the data, this is the data file, and it will estimate the richness. Or you can use the vegan, and for activating the vegan, just command, give this command library and vegan, and the function of vegan will be available. And with this vegan, you can estimate the richness and Simpson index and Shannon index and paleo evenness. And you can also have the plot using bar plot, and you can also estimate the correlation between Shannon-Weber index and Simpson index. And I also suggest to see the function of NOSIM. This is very also useful. And here are some outputs I have shown here. Another package, this is also very commonly used, and this is more user friendly because this is the window base, and just through the menu driven, you can estimate different kinds of indexes. We call biodiversity R. And for installations, there may be some difficulty. Therefore, I suggest to follow strictly the step given in this website, in this site. After, if you properly install, then you can run the R package in R Studio, and the new window will pop up. And using this new window, you can estimate different kinds of parameters. And for this, for loading biodiversity R package, just write this library biodiversity R, and then biodiversity GUI, and this will show the interface window, and you can easily run the program, and you can also import the data. Yeah, this is the R. After, you also need the R commander, and within R commander there is one menu. You can see here biodiversity R, and if you click here, you can see different kinds of options like other menu based software, and there are many functions that you can play around. And after clicking this, you can also see here what types of analysis you want, what types of data, and where you want to output like that. This is the simple, and if you play for few parameters, then you can estimate other parameters as well. After getting the outputs, interpretation is the major parts. I'll hear briefly what is richness. Richness means the number of species or land masses or even particular traits that may be at household or community, sites or landness that quantify how many different types the data set has. And Shannon index, also called Shannon Diversity Index or Shannon-Weber Index, includes both species number and evenness, where the greater number of species increase diversity, as does a more equitable distribution of individuals among species. High Shannon-Weber Index is a representative of diverse and equally distributed community, yet it is strongly influenced by speciesness and by rare species. And Simpson Index, it measures the diversity, which takes into account both richness and evenness. The value of D range from 0 to 1. The greater the value, the greater the diversity. The Simpson Index gives more weightage to evenness and common species. And evenness, also called Pelu evenness, is a measure of the relative evidence of the different is considered to be less diverse than one in which several different species have a similar evidence. Its value range from 0 to 1, and 1 is complete equitability. Yeah, these are some mostly my publications that have some details, explanations of this some parts that I shared earlier. It is also uploaded in the website of Grassroots Institutes. And these are some information for further readings. There are some also detailed explanations. These are some web links that are very useful to get started. For example, you can read art tutorial, quick art. There are many very simple and very stepwise explanations, and many students or professors can follow this one easily. And these are the software that I suggest to try using for estimating the agrobiodiversity, and these all are free. If you are familiar with RStudio, and you can easily run these two vegan and biodiversity R. Past is a simple, very simple package. It's not a R package. You can run differently without installing RStudio. And in the past also there is different kinds of diversity indexes. Yeah, thanks. I would like to thank especially Dr. Hasrat Arzumen, the Grassroots Institute, and all partners and all of you. I am very happy to share briefly about this possibility. And with this different kinds of option I provided here, anyone can estimate different kinds of diversity and score at different level, household level, village level, or district level, or even country level. And we can compare the different sites or different household so that we can target the collection mission, or we can also use for gap analysis. If you have any questions, you can ask either through chat box or email that already Dr. Hasrat shared. I'd like to thank you. That's all for this presentation.
Thank you, Dr. Joshi, for this presentation on agrobiodiversity measurement, and for your information provided on various tools to calculate indices and to analyze data, as well as with regards to softwares that may be used to appreciate on qualitative or quantitative basis the agrobiodiversity at different levels, as you just explained. So thank you very much on behalf of all the attendants for this information. And now